Here we have Hershey and Chase conducting their experiments. Stay tuned for the interview.
It is an extreme honor to have Alfred D. Hershey and Martha Chase on the show today. These two scientists have challenged the long-perceived notion that protein was the carrier of genetic information. Yet, in 1942, Avery McLeod McCarty concluded that DNA was the carrier of genetic information. Could you explain why you hypothesized that the protein was the carrier of genetic information?
Absolutely. First, I must say that we weren't the only ones in the scientific community to believe that the protein held genetic information. The protein is a complex structure that can be folded to form primary, secondary, tertiary, and quaternary structures. Proteins are also enzymes, which are vital for our survival. We believe that the genes in DNA that was found in bacteria doesn't have the same function and composition as more complex organisms.
Interesting. I can see how the scientific community would be divided on this topic. Can you elaborate on some of the predecessors in genetics besides Avery McLeod McCarty that influenced your experiment and hypothesis?
Well, we are fortunate to have been the benefactors of knowledge that previous scientists have contributed to the field. Mendel is the backbone for all scientists studying inheritance and genetics. T.H. Morgan and Frederick Griffith are also extremely influential. T.H. Morgan discovered genes exist as part of chromosomes, nucleic acids, and proteins. Frederick Griffith experiments on if genetic traits can be transferred between different bacterial strains opened the floodgates of the study of genetics.
Don't forget Felix Durrell. He found bacteriophages, or phages for short. Phages are cells that engulf bacteria. This property makes them ideal to study molecular genetics. They consist of a phage head that holds DNA, or sometimes RNA, tail sheet, and tail fiber.
Wow. It's incredible how far science has come and the wealth of knowledge you two have on the subject. Now let's get to the meat and potatoes of the experiment. Could you explain to me and the public your experiment?
We wanted to see what was the genetic material of phage called T2. Was it the protein or the DNA? T2 is one of several phages that infects E. coli bacteria. Once T2 infects E. coli will eventually rupture and spread the T2 copies. First, we had to be able to differentiate what parts of the virus were passing the genetic materials. The phage were grown with radioactive isotopes of sulfur to tag the proteins in the first batch and phosphates for the DNA for the second batch.
We mixed the phage and the bacteria together. The phage would then infect the bacteria. Once the bacteria was infected, we agitated the mixture of each batch in a blender to get rid of the phage parts outside the bacteria. After agitation, centrifuge the mixture, so that the bacteria sinks to the bottom to form pellets, and the free phage and phage parts remain suspended in the liquid. Finally, measure the radioactivity of the pellets and the liquid. Our results revealed that the radioactivity remained outside the bacteria in batch 1 with the radioactive protein. The second batch revealed that the radioactivity was found within the cell. Also, the bacterial cells in batch 2 released new phage that contained some radioactive phosphorus.
Our conclusion is that phage DNA enters the bacterial cell, but the phage protein does not. Therefore, the DNA acts as the genetic material for the phage.
So, your hypothesis was wrong and Griffith was correct about the DNA being the transforming agent in his experiment?
Yes, our hypothesis was incorrect and Griffith was correct about DNA being the transforming agent. However, our experiment proved that Griffith was correct, that the DNA is the genetic material, not the protein. With this new understanding of DNA, the scientific community can gain new knowledge about the structure of DNA.
Makeup of DNA.
Alright then. Thank you both for your time and contribution to furthering the scientific understanding of the complex molecule of DNA.
Thank you guys again for the interview. Up next, can women truly do a man's job?